Welcome to Ask the Expert. This is a brief, informative, lively discussion with experts in type 1 diabetes and related interdisciplinary research. We are going to be recording this event and we'll post it on the Sugar Science site YouTube channel shortly after the presentation. If you have any questions for our guests, please feel free to enter them in the chat or raise your hand at the end of the presentation. And today we have as our guests Dr. Alexander Hoffman coming to us from UCLA. Here in lovely Los Angeles. And he is the Thomas M. Asher Professor of Microbiology and Immunology there at UCLA and the founding director of the Institute for Quantitative and Computational Biosciences, the QC Bio. The Hoffman Labs really focuses on developing a predictive understanding of the cellular responses to pathogens and cytokines. And I'm sort of, reading from his bio, but quantitative experimentation is iteratively coupled with computational modeling to derive insight and develop hypotheses. And the Hoffman Lab models immune and inflammatory responses, and that's provided some key insights into how these complex regulatory networks function, which is sort of aimed right at the heart of what's happening in the autoimmune dysfunction of islets. And so we're going to discuss his new paper, which is a very, very cool paper. And I'll let him, introduce that. And also like what they're doing in the lab and how it might have overlay and impact on the type 1 diabetes field. So welcome. Thank you so much, Dr. Hoffman.
Well, thank you for having me. I'm really excited to learn more too.
It's great for you to join us. And I wondered if you could give us just sort of a thumbnail sketch of your career path. What kind of excites you most about the research you're doing right now? And of course, let's talk about your paper.
Yeah, no, I'm very happy to be here and learn more. That's what's great about doing science, that you are always able to interact with new people, new collaborators, and you learn more ideas. My own trajectory really began because I was excited about actually sort of, as naive as it sounds, trying to understand the universe as a physics student, as an undergraduate in physics. And
I like that.
And what strikes me, and I think it is relevant a little bit, is that when you look at the universe, it looks like it's a static thing, but then over time you realize it moves. Everything moves, and there's a lot of motion. And I think in biology, and I transitioned to biology shortly after that and did a PhD at Rockefeller University in biochemistry. It's sort of similar that most of our measurements that we do in biology are really a snapshot of what is going on. We measure mRNAs, we measure proteins, we measure cytokines, we measure the presence of cells. But it's always just a snapshot, and that really does not tell you about all that movement. And of course,
yeah, that's really perfectly put. It's a movie, and we've just got the snapshots.
That's right. Yeah, and so that's what also then began when you take snapshots, at different time points. Then you realize, well, if you want to represent what's really there, you need to fill in the time points that you haven't measured. You have to fill in that movie, all those frames, because, and so that's why I got interested in using computational modeling, because with modeling, you can simulate and you can take as many time points as you want, even if you only measure one time point every few hours. You can then infer what happens in between when you have a little bit of an understanding.
Right. Fill in the blanks.
Fill in the blanks. And so that's what, even though I started off as an experimentalist, that's what got me interested in using computational models to then interpret and extrapolate from the measurements that we can do. And so that's why I'm here. And UCLA, we have a fantastic group of interdisciplinary scientists from different departments. And of course, they're working on all kinds of different things. But one thing they have in common is this interest in using mathematics in the service of biological questions. And,
yeah, I love the intersection that you've got there. The Institution of Quantitative and computational biosciences, and I'm sure that has a sort of long reach into many different disciplines.
That's right. Yeah. Yeah. So my own interests have been in inflammation and immune responses, which of course are really dynamic because they typically have a starting point, right? The pathogen enters the body, the injury happens, and then there's a cascade of events that occurs. And that ultimately, when things go well, result in resolution, where you're almost getting back to where you were. There might be some memory of that injury, but there's a lot of dynamic things that happen. And so that's been our interest in the lab. And so this new paper describes how the dynamics of signaling in response to pathogen exposure might result in epigenomic reprogramming in a change in the epigenome, and that provides a level of memory of that event.
Yeah, and so let's just sort of give it a shout out for people who haven't seen it. It's found in Science, and it's June 18th, 2021, volume 372. The title is NFK beta dynamics determine the stimulus specificity of epigenomic reprogramming in macrophages. So yeah, let's dive into that a little bit.
Yeah, well, in the paper we describe how pathogen exposure can lead to epigenetic reprogramming, but we find that not every pathogen stimulus produces this epigenomic reprogramming, but there's some specificity. And so that's actually exciting because that means the memory that the macrophages have of their pathogen exposure is very specific. It's specific to the pathogen. So the memory is pathogen specific. So that's great. But what was really unclear is how that might be the case, because every pathogen activates the same transcription factor, the same signaling pathway. So if you activate the same signaling pathway, how can you have different epigenetic memory?
Right. Yeah, that's a huge question.
Yeah. So I think what we discovered is that even though the same factor is activated with different dynamics. And so it's just like having a telephone wire or having a wire. You can use it to turn a light on and off. Great. Or you can use it for telephone conversations. And so then the single wire can transmit different information because the voltages or the electricity is temporally modulated, it's dynamic, right? So
you're almost talking about like a code.
That's right. That's exactly it. And so there's a code, and so it's the code by which a macrophage communicates with the nucleus, right? Where the outside, the nucleus where the genes are located, are shielded from the outside, but there is a signaling pathway that connects the nucleus to the outside. And so that signaling pathway represents or uses a code that is dynamic to make very specific changes. And so we've been interested in trying to understand how this code works and what the code means. And so I could show some slides of that and illustrate it.
Yeah, that sounds great. Let's see a few slides just to sort of a walkthrough.
So I'm just going to. Okay, so. So I think we've already talked about this, but I think here you should see a little movie of cells. These are macrophages in a dish, and you see that they're exposed to a cytokine called TNF, and they respond in a way where this transcription factor, which is labeled fluorescently, response in an oscillatory manner. You can, if you look in detail, you can see how the transcription factor goes in and out of the nucleus. But if those same macrophages in a separate dish are exposed to a different stimulus, in this case endotoxin, the responses are much less oscillatory. They are, of course, every cell responds slightly differently, but these are non-oscillatory, and on the left they're oscillatory. So wow,
so that's cool. This is a very cool video.
Yeah, it's a video that that I've always loved, and then I think actually Francis Collins put it on his blog as well, so I was excited to see that.
That's a nice little nod.
Yeah. Right. So nice. One way to to describe then is that here's this kind of heat map where you see about 400 or 500 different cells, and we show here in red when NFkB, the transcription factor, is in a nucleus, and you see that every cell sort of oscillates. They're not synchronous; they're kind of out of sync, but everyone oscillates. And here on the right, most of them are non-oscillatory. And so we can make measurements of hundreds of cells in lots and lots of different conditions, which means that we can generate, lots and lots of heat maps just like this. And then we can try to figure out, well, what do they have in common, and what's distinct? And
their signatures, you're kind of trying to drill down on their signatures, right? Right.
What are the sort of dynamical features, if you will, that say something about the stimulus? And if we find what the features are that are specific for a stimulus, then that represents in some way a code word or an informative feature, right? So in the case of TNF, it's actually these oscillations. And so that sort of statistical analysis of all of this crazy amount of data led us to identify 6 what I call signaling codons, or these are just like a genetic codon represents a unit of information about a genetic code. These signaling codons represent a unit of information about immune signaling.
Now, just for a second, I'm pretty familiar with TNF, CpG, LPS, but what is P3C4 and poly-IC?
Yeah.
What are those?
Sorry, yeah, poly-IC is single-stranded RNA, represents, it's a compound that is analogous to or is used in place of a single-stranded RNA virus. And so you see that in response to these different compounds here, right, these codons are deployed specifically. So there's high speed in response to these compounds and slow speed here. Only TNF oscillates, the other ones don't. Total activity varies, is highest with LPS, and there's a couple of other ones. These ones all look the same, but actually if you use different concentrations or doses of these, then the peak amplitude changes. So from the statistics, we find that all of these 6 basically represent code words in this code.
Yeah.
And then we thought, well, if they represent code words for this code, then we should be able to teach them to a computer and do machine learning. So we teach these codons to a machine learning algorithm, and then it turns out the machine learning algorithm can identify with, if we present it with some signaling trajectories, then a machine learning algorithm can identify what the stimulus was that the cell was exposed to. So
that's very impressive.
So that's kind of similar to if you're a gene in the nucleus and you're seeing transcription factor dynamics, you need to know what the stimulus was that led to that, and you need to know whether to respond to that or not. Did it come from a virus? Did it come from a bacterium? And if I'm an antiviral defense gene, I need to be induced only with the virus, not with the bacterium, right?
Right.
And so that's what this does. And so we can show also using this algorithm where the cell might be showing some confusion. So for example, so here we see the predictive accuracy, if you will, of each ligand. But we also see that in some cases there's confusion. So for example, PAM3CSK is sometimes confused with LPS. And that's maybe not a surprise because actually both of them are bacterial ligands, right? So actually, maybe the confusion isn't necessarily bad. It's just, you know, there's no need to distinguish PAM3CSK from LPS that well. So anyway, that's healthy macrophages. But then we thought, okay, how about macrophages from a mouse that models Sjögren's syndrome. So Sjögren's syndrome, I think you're probably familiar with this, an autoinflammatory disease, or an autoimmune disease. And interestingly, it flares up sort of sporadically, right? It's not always chronic. It's not really persistent. It's flares. And so when we take macrophages from this disease model, and we look at how these signaling codons are deployed in response to TNF, LPS, and poly-IC, we see actually that these macrophages do just as well for most of them. But here's one that is completely different. And this one is the same. And this one is also very poor. And this one is the same. So there's actually only two that are misregulated, and the other ones are doing perfectly fine. And so the misregulation of these two then results in much more confusion, such that typically TNF and LPS are not confused very much. But in the Sjögren's macrophages, they're often confused, which means that if the cell is seeing TNF, but it responds as if it saw endotoxin. And so a TNF comes from a host, from other neighboring cells, right? But LPS actually comes from a bacterium. So if you're confusing TNF and LPS, that leads to much stronger responses that are totally inappropriate.
So is this kind of saying that some kind of bacterial insult could lead to the wrong immune response?
Yeah, it actually says that these cells, these Sjögren's macrophages, have a tendency to respond to signals, host signals, from neighboring cells, as if they were exposed to bacteria. So even when there's no bacteria around, they respond as if bacteria were present.
So interesting.
So we think of we're thinking of these types of autoinflammatory diseases now that some of the etiology might be this kind of confusion, where maybe we can think of them as confusion diseases, where signals are confused. So that's what I want to leave with you, that when we're thinking about autoimmune disease, and maybe that's where we're hoping to understand the etiology of type 1 diabetes, maybe the immune system that has this destructive effect on the pancreas, maybe thinking about that the cause is some sort of confusion of signals.
Yeah. And I mean, I just, if you wanted to sort of now shift gears and say, let's replace Sjögren's syndrome with type 1 diabetes, how would you set that up?
Yeah, so that's what I'm hoping to learn from you. But,
I don't know about me.
Right. I mean, so here we're looking at macrophages, right? And macrophages, of course, are the initiators of immune response. In the end, it's probably cytotoxic T cells that play a key role in the destruction of
the pancreatic beta cells. Yeah,
pancreatic
for
beta
sure.
cells.
But I mean, there's several players there. I mean, now some people are pointing fingers at the antigen producers, the B cells, but there's definitely, the CD8s are there. And the antigen presenting cells play a role. I mean, the whole, it's kind of funny. I sort of like sometimes refer to this as a bad ballet, right? So it's like all the players are not quite doing their correct jobs, but it is, macrophages can be one of the first ones on the scene. So that is why it's really interesting to think about what they're interpreting from that scene. And it always goes back to, is this, murder or suicide? It looks like more and more data is surrounding the fact that maybe the beta cells are, inherently dysfunctional. And then that the immune system, is now responding to some dysfunction. Although there's another camp that says the cells are actually senescent and that they're just not getting cleared, and and so the immune system is responding to that, or not responding enough, basically, to that. So it's a real, there's a lot of hypotheses sort of out there now, I think. But one thing I wanted to bring up is I think that this mathematical modeling that you're doing is, is really fascinating, because you can kind of go one by one, just like you showed us with the Sjogren's syndrome.
No, that's really exciting. Yeah, we should. So the heterogeneity of what cells expose and what cells do, and I think certainly for apoptotic signaling and cell death signaling, there's some very good math models that account for some of the heterogeneity. Typically, that sort of modeling has been done in response to chemotherapeutic agents in the context of cancer.
Yeah.
But I think that could certainly be applied to, to the process that you described in pancreatic beta cells. So that's another.
Yeah, there's so much work to be done. It's it's unfortunate that a lot of the funding is actually being drawn away to type 2 diabetes because the market is so much bigger. But I think essentially the questions that can be answered in context of type 1 diabetes have a lot of far-reaching, impact on other autoimmune diseases and actually the functionality of the immune system itself. So that's kind of, it's a real frontier.
I would welcome any comments and interests to collaborate or otherwise. So thank you so much.
Fantastic. Thank you so much. I love that.
Great.
Thanks again.
Thank you. Until soon.
All right. We'll talk again soon. Bye-bye.